Couple of quick announcements today. The first one is a blatant plug for Journal Virology. My virus is on the cover. Yay. Second one is that the seminar speaker this week in the biology seminar series is a fellow by the name of David Wiesler from University of Washington. I think he's going to be talking about really cool virus structures. I'm actually not sure because he's done all kinds of other interesting structures, but the virus ones are the most interesting. And so with any luck, that's what we'll be talking about. So that's on noon on Thursday, and I will remind you again on Wednesday what's going on there. Wednesday, we have a guest lecturer, Alec Hirsch from OHSU, who's a real expert on flaviviruses. And so we'll be talking flaviviruses then. I will ask him if he's willing to have his lecture recorded. I'm not sure if he's going to do that or not. So I recommend being here on Wednesday. Should be a lot of fun. So that being said, like to get through the PicoRNAviruses today. We'll see where we get. Yeah? Will his lecture be on the exam? Nah, I figure stuff we talk about in class. No, yes, it will be, definitely. That material will be on the exam. I will be sitting back here somewhere and taking notes. And from those notes, I will be putting together exam questions.
So will those exam questions be somewhat presented in the review?
Will those exam questions be somewhat presented in the review? The review is not like for weeks. I don't know what I'm going to have in the review. But I will have his slides, and I will potentially use some of those in the review if I think it's something that we should talk more about. Yeah? Yeah, so I will definitely try and post his slides. That's usually not a problem. I have last year's, which are already posted, and I suspect it'll be similar, although the society's probably going to do more Zika and less West Nile. Okay, other questions about Wednesday's lectures? Okay, so just really quickly finishing up on the plant viruses because I didn't get to this last slide last time. We talked about, again, these are single-stranded RNA viruses, like we'll be talking about for the rest of the lecture today. Bromoviruses, this is just, plant name, what the phenotype is, et cetera, viruses. The really fascinating thing, I think anyway, about these viruses is that they've got these, individual capsids, each of which has a different segment of the genome, and so you have to be infected by all of them. You have to have, at least in the case of cucumber mosaic viruses, 3 virions with 3 different nucleic acids. How many went through and figured out how frequent that is? Any of you? No? I'm not going to ask you a question about it. And then these movement proteins, again, because you've got to move these individual virions around the plant, and plants are really good at protecting from cell-to-cell movement, which is why the viruses have evolved ways to get from cell to cell. And then the rest of today, we'll finish up talking about tobacco mosaic virus very briefly, but again, it's a pretty big deal as far as these viruses are concerned. Yeah?
I think it's a 1 out of 9.
1 out of, so that's not quite right because you have to do the Poisson distribution. So it actually ends up being slightly less than that. But yes, close to 1 out of 9, but less than 1 out of 9. So, and if you'd like to, I can go over your calculations if you like. I'm happy to do that another time. But instead, I'll ask you a much easier question for the first clicker question of the day, because everyone's clear on our viruses, right? Except I need to start. Forgot to start my clicker. That was silly of me. I'll just show you the cover picture of the Journal Virology again, just to show off a little bit more. Go, go, go. Yes. Eventually. Good. Okay. Let's go back to showing this now. Too much technology going on here. Thermotolerance of Dicanthelium lanuginosum is due to fungal infection, plant virus, fungal virus, air pressure, A and C. That was last week. I had a weekend in between. I can't remember any of this stuff. One of the coolest things we talked about all term, I think anyway. So we're going for 100% for every single question today, right? Pardon? 10, 5, 4. Okay, it is 100%, except for the fact that not everybody can agree that it's both of them. So they have to be infected with a fungus, and that fungus has to be infected by a virus. Otherwise, they cannot survive at high temperatures. So wanted to talk really briefly about tobacco mosaic virus. Again, we're not doing this justice by any stretch of the imagination. There's this whole book here that I have up front about these viruses. And one of the things that I wanted to mention is that, yes, it's the life of a virus, tobacco mosaic virus. Let me turn the lights on here. But most importantly here, so what I wanted to show you was this apparatus. And so you remember how the original definition of viruses was done? It was about going through these really small filters. And so this was that kind of filter that was being used in those very early virus experiments. So this particular filter blocked all bacteria, so nothing would grow down here at the bottom. But you could still have this disease-causing agent that went through these whole filters. So now we can switch back to here. This was Contagium, vivum, fluvium. So again, it was a liquid because it went through these filters, but still caused disease. And as true with many of these plant viruses, originally called tobacco mosaic virus, and it does infect tobacco. I've heard rumors, but I've never checked this. Apparently, if you just go to cigarette butts and take out that last little bit of tobacco which is there, you can isolate tobacco mosaic virus from most of these. But it turns out that it also infects tomato, and this is the example of tomato down here at the bottom, where you have pretty serious infection. Here's a non-infected plant here, and then these are various infectivities that happen here. This is what that particular virion looks like coming through the filter. And this was all done by Beijerinck. I'm not quite sure whether I'm pronouncing that properly. Dutch researcher. And I just love this quote because I wish I could get away with something like this in my papers today. You know, I was therefore able to carry out a series of incontestable infection experiments, the results of which I shall briefly describe. And his paper's about 48 pages long. So, and the whole thing is here. It's actually really fascinating. And there are a couple of things, so I'm not forcing you to read all 48 pages of it. Here, he also uses the term virus, so it's not just contagium, vivum, fluvium. So I think he was really the first one to use this term, virus. Can only grow in growing plant tissue. So we already had this idea that it has to be this infectious parasite. There's an infection process that's actually involved. And this was in 1898, which is really pretty amazing as far as I'm concerned. And he also showed that this infectious agent was not only really small, but heat and drying stable, which is why you can probably isolate it from cigarette butts. In fact, I sent an email down to one of my buddies at Oregon State and said, you know, could you send me some tobacco mosaic virus? He said, yeah, just find some cigarettes. You'll find it there, which we didn't do, but we thought about doing. So that was, again, the original virus discovery, whole book on all about how to work with it. But it turned out to be a really wonderful model system for thinking about viruses and actually even thinking about viruses being alive. So TMV was crystallized, and in fact there's some pictures of crystals in here. I won't switch over to show them to you. The virions could be crystallized. And this was one of the really first indications that, hey, you know, you can't crystallize life, so therefore this can't be alive. This was sort of the explanation at the time. That was in 1935. Wendell Stanley was the one who did this crystal structure. For a long time, they thought that it was just protein, and there was no nucleic acid that was involved with it at all. Turns out there's a lot more protein than there is nucleic acid that we'll take a look at in just a second here. He got the Nobel Prize in 1948, and this picture right here, any of you recognize it? Probably not. Stanley Hall, UC Berkeley, and I spent 6 weeks of my life working in the lab right up here in this corner. So this was Stanley Hall. Berkeley has a lot more money than PSU does. So this is what that building now looks like. Renovated completely in 2007, and I wouldn't recognize it. But it's basically the building that TMV built as far as I'm concerned here. So what does that look like? This is the structure of TMV. We may have a chance to go and take a look at this image a little bit later on, but this is the real classical helically symmetric structure. There's an RNA, which is here in red, and the vast majority of this structure are proteins. Here in just two different blue colors. They're all identical. It's a single capsid protein that looks like this. What's the big difference between this capsid protein and all the other ones we've looked at so far? It's all helical. No beta strands, beta barrels at all that are associated with it. So all of these are a little bit different. You can also see this little loop down here at the bottom. That's where the RNA goes. The RNA actually sits down in this little loop structure, and the whole thing wraps around it completely. Yeah, Ian?
So the light blue and the dark blue are just to show the helical nature of the capsid and not to differentiate that these are different proteins.
Yeah, exactly. So the light blue and dark blue are just to differentiate the different capsids, which are all, again, identical, and they're this structure. Is this why they were able to crystallize it? Turns out you can crystallize a number of virions. One of the main reasons that this one was able to be crystallized quite early on was because you could get massive amounts of it. And just by infecting a few tobacco leaves, because it has a nice movement protein, it can move around through the whole plant. And you can get a billion virions actually really quite easily from one of these tobacco mosaic virus infections. And a lot of tobacco is infected with it, which is, again, why you can go to cigarette butts and find this. OK, so that's all I wanted to talk about as far as plant viruses are concerned. And again, my apologies that we cover plant viruses in about an hour. We could, again, easily spend a whole course doing this. And if you're interested, look at some of the courses at Oregon State. They do a really nice job with all kinds of different plant viruses, et cetera. So any more questions on those RNA viruses from plants? Now let's switch over to animals. These, again, are single-stranded RNA viruses. Most of the people who work on them call them PicoRNAviruses. I really don't like that pronunciation. And the reason is that I really think of these as PicoRNAviruses, because they're really small RNA viruses. Where the PicoRNA thing came from, I have no idea. So talk a little bit more about just RNA viruses in general, but mostly concentrate on polio. Now we're talking about human disease-causing viruses. So we'll talk a little bit about pathogenesis and control and those kinds of things for those people who are really excited about that. I see a couple of you. And then much more about the interesting stuff from my point of view, which is the molecular basis of that. And some of the really important aspects from the molecular point of view, which actually turn out to be important for treating disease as well and understanding how these things replicate, have to do with entry, an IRES, which is an internal ribosome entry site, and then replication, which is always going to be an issue for these RNA viruses, is how do you replicate and translate, just like we talked about for MS2 and Q beta earlier on. So where do they come from? When we talk about these animal viruses, where do they come from? Often is going to include disease. Yay, excitement. Structure, we've actually already talked about these picoRNA virus structures already. Classic pseudo T=3 structure. The way those get put together is actually really quite interesting, because it turns out these are all made as polyproteins. and then chopped up by various proteases in order to put that structure together. So it starts out as one big protein and then gets chopped into smaller proteins. How these guys get in, it turns out that these, the PicoRNAviruses, actually kind of analogous to a lot of the bacteriophage. So there's a conformational change that happens in the capsid, and then the genome is released all with the plasma membrane. So really quite different relative to the endocytosis, which is what we normally think of when we think about animal viruses infecting. Genome, big picture with the genome is that it's this single RNA that encodes a single protein, which is really bizarre. Now, what does that single protein do all the things that a virus needs to do? And again, it's because it gets chopped up a little bit. Replication and translation, again, RNA viruses have this problem that you have to deal with replicating and translating trying to happen at exactly the same time. And the way the viruses get out is actually really pretty straightforward. PicoRNAviruses. The best studied of these, for obvious disease reasons, are the polio viruses. These are enteroviruses, entero meaning that they're in the gut. But poliovirus causes this neurologic disease. How the heck does that happen? The vast majority of polioviruses actually are an enteric virus. And the actual poliomyelitis, which is the disease, happens at a very, very low frequency. And so many, many people can be infected with polio and have no symptoms whatsoever. Spreads actually extremely well from fecal to oral and really having to do with contaminated water. There are other viruses, Coxsackie virus, which is a great model system to study for polio because it really doesn't cause that much in the way of disease. And then rhinoviruses, these are probably the most common. Your common cold, rhino, the nose, that is probably the main reason that you have a cold. Or if someone says they have the flu and they're actually telling you that they have the flu, they probably have a rhinovirus infection. Highly unlikely that it's the flu. And we'll talk more about that later. But it turns out that there are lots and lots and lots of viruses that are related to these polioviruses, et cetera, and all small RNA viruses, PicoRNAviruses. And here in our table in the textbook, it says 4 additional picovirus genera, PicoRNAvirus genera that are not listed here. There's probably way, way, way more than that. And when you do the metagenomics, we talked about a little while back for the taxonomy, but also for single-stranded RNA viruses, there are really massive numbers of these small RNA viruses with very similar kinds of genomes and replication strategies that are present all over the place. Yeah?
Does having a single protein that is cleaved do anything to conserve space versus having multiple proteins that are translated separately?
Yeah, so the question is, basically, I'll back up your question a little bit. Why have a single protein? We'll talk more about that when we talk about the genome in just a second. Yeah?
Is poliomyelitis a specific strain, or is it a different species than the enteric?
Great lead-in. Thanks, Ellis. So we'll talk about this in just a second here. So again, rhinoviruses, common cold, but polio is the best understood of these. Usually, the poliovirus, again, it's an enterovirus, so it's in the gut, has nothing to do with neural disease. So the vast majority of polio infections don't cause poliomyelitis, which is the neural disease. There are a couple of cases. So when you do have polio that gets into the nervous system, 1 in about 200 of the people who do get it into the nervous system. The reason that this is a underestimate probably is that most people don't even know that they're infected with polio. Just goes straight through and there's no effect whatsoever. However, if it does get into neurons, about 1 in 200 of these people are paralyzed and 5 to 10% of those die, mostly in young children. And probably this has to do with the fecal-oral transmission route. Any of you who have kids know all about what happens to kids as far as that's concerned. One of the best poliovirus vaccines, the so-called Sabin strain, which we'll talk a little bit more about later on, doesn't replicate in neurons. And so that process means that it is only going to be in the enteric state, but it turns out it gives a nice immune response and can therefore protect against the viruses which could potentially replicate in neurons. And it turns out that the change in this strain is in this internal ribosome entry sequence. So again, we'll talk more about that a little bit later on. Poliomyelitis, Alice's question. This used to be a really, really major problem, and the usual manifestation is partial paralysis. And very often in legs, and so this is an example of a child with poliomyelitis in one of their legs, and there's also an example from 5,000+ years ago in an Egyptian stele, sort of a grave marker, where it looks as if this guy has had poliomyelitis as well. So probably poliomyelitis has been around for thousands of years. Now, whether this is actually true or not is not entirely clear. Used to be tens of thousands of cases of polio per year in the US before the 1950s, which is when America was supposed to be great, but we won't talk too much more about that. So lots and lots of cases in the US of polio. One of the most successful vaccines, probably next to smallpox, has been the development of the polio vaccine, originally by Jonas Salk in 1955. So that's about 60 years ago now, where he, and I say he, of course, means a lot of workers that were working with him, purified very large amounts of polio virus, but not from people who were infected with it, but from cell culture. Any of you have seen the HBO film Immortal Life of Henrietta Lacks yet? I highly recommend doing that. It was probably because of those cell cultures that the development of this vaccine was possible. The HeLa cells, exactly. So it turns out that HeLa cells replicate poliovirus extremely well. Yeah?
Can you repeat the name?
Which name? Yeah, The Immortal Life of Henrietta Lacks by, well, the book, which is a fabulous book, Rebecca Skloot from Beaverton, I think, originally. So a local author. But it's a fabulous book and apparently a really good film. I haven't seen it yet. But Oprah is playing one of the main characters. So got to check it out. Pardon? And it's on HBO. Yeah, it's an HBO-specific thing, so which I don't have HBO, so wait till it comes out on DVD. That was really the impetus and what allowed this whole process, in fact, to take place, was this development of growing viruses in cell culture, massive amounts of them, then inactivate that virus, and then use that to inoculate people. And because of the development of this vaccine, literally 2 years later, there was about 90% reduction in polio in the US. Just absolutely amazing progress that happened, and then by 1979, it was completely gone. There was ever a problem with this polio vaccine, and that was you had to make huge amounts of the virus in order to do these injections, needles. It's a problem particularly in places outside of the US. So there was another vaccine that was developed called an attenuated vaccine. And we haven't talked about attenuated vaccines, have we? No? Everyone clear on attenuated vaccines? Answer questions on exams about them?
Please no.
Well, so I'll talk about it now, so I can ask you questions about it. So the idea of an attenuated vaccine is basically it's a weakened virus. And in this particular case, the Sabin vaccine is a weakened polio vaccine. And so how do you get a weakened vaccine? What you do is you do multiple rounds of infection. So you infect something, and in this case, it was a cell culture, if I remember correctly. You infect that cell culture with the virus, you take that virus, you infect it again in that cell culture, and you do this multiple times. This was originally done by Louis Pasteur for rabies virus, and each time you have the virus infect, it ends up being slightly lower pathogenesis, and so causes less disease. And that makes sense. Again, these viruses are completely dependent on their host. If you're causing a lot of disease, then it's probably not that good for the virus's point of view. And this is nice because you can do these attenuation processes in a much faster way than it would be infecting someone, getting those particular pieces back out. So cell culture is really important, and also animal studies that were done for this. And so in that process, the oral polio vaccine was developed, which is now a live attenuated vaccine. So it's still functional as a virus, but as I showed you before, it doesn't replicate in neurons. But it has one really big advantage relative to the inactivated polio vaccine. And that is that this oral polio vaccine can replicate. It can make more of itself. And so if you immunize one person or one kid, they will then produce this virus in their gut because it's an enterovirus. That ends up in the fecal material, which can then, if you have fecal-oral transmission, which is the main problem with getting polio in the first place, now you can actually basically spread the vaccine in the same way that the infection process would otherwise be taking place. So most of the immunizations now for polio, particularly in the developing world, are this oral polio vaccine. Turns out there's some problems with the oral polio vaccine that we'll get back to a little bit later on. So because of these really wonderful vaccines, there's been this project called Polio Eradication, and we are getting close. We're getting really, really, really close to completely eradicating polio. There was a start of this was, again, I'll back up a little bit, nearly 500,000 deaths per year in the mid-20th century, the vast majority of them in the developing world. So in the late 80s, they decided, okay, we're going to try and get rid of polio. Starting out then, it was still a pretty large number. In 2015, there were 74 cases, which is really pretty darn amazing that we've gone from 350,000 down to 74 cases. It's cost over $2 billion, which may sound really expensive, but if you think about the number of lives that were lost, it's actually a really, really, really good investment. So I wanted to spend a couple of minutes talking about this whole effort here. So this was the case in 2009 and 2010, where all of the poliovirus cases were, and you'll notice that they're in definitely developing parts of the world. A couple of things to look at here. There are three main types of poliovirus circulating. There's type 1, type 2, and type 3. Again, ridiculous creative names here. But you'll notice that the vast majority of cases here are 1 and 3. In fact, there haven't been any type 2 wild type infections that have taken place in the last, I think, about 20 years. So this was 2009, 2010. Fast forward 4 years to 2014. Now we're down to just a few places, Iraq and Syria, and Pakistan and Afghanistan, and then a couple of places in West Africa, particularly Nigeria. Fast forward again to, when was this? Last night. And the numbers for poliovirus for last year and this year, the only places which they are occurring are in Pakistan and Afghanistan, and a couple of cases in Laos. Now, these are really interesting cases here. These are what are called CVDPV. What the heck does that mean? So these are vaccine-derived polio cases. So as I mentioned before, there's a problem with the oral polio vaccine, and that is that it's a live virus. And in this attenuation process, what has happened? Mutations have arisen in the poliovirus genome. Well, at a low frequency, it turns out that those mutations can revert. They can go back to the wild type version, which of course can cause disease. And that's what's happened in Laos, in Southeast Asia. Couple of very isolated cases, but these are clearly vaccine-derived poliovirus disease. That vaccine is based on type 2. And so it's really easy to tell if that is a vaccine-derived case or a wild-type case. But this is still very stubborn, hanging out here, and these are These particular regions of Pakistan here next to Afghanistan are also known as the tribal regions. They're not a terribly safe place to go and vaccinate, which is one of the major issues with this. Yeah?
If we were to discontinue this project, would poliovirus return?
Oh, so the question is, would poliovirus return if we stop the eradication project when we still have some cases? The answer is those would definitely spread, and we have some examples of that. In fact, I'm going to take that's on the next slide, so I'll get to it in just a sec. Yeah?
Are there animal reservoirs?
Are there animal reservoirs for polio? No, there are no animal reservoirs for polio, which is one of the reasons that we can actually do this whole project in the first place.
What are the types characterized by?
The types, so type 1, 2, and 3, actually are antigenic types. So it's just what kind of antibodies you have that are interacting with this. This is a serological definition. We also know the molecular definition for that too, but this is all a serological definition. Yeah, so let's take a quick look at what happens here in the case of stopping this. So Nigeria is a really nice example of how this immunization process has worked very, very well. So 2013, 53 cases. 2014, 6 cases. 2015, none, and people stopped. And then 2016, it came back. And so this process, you have to really keep going for, and what the WHO recommends, the World Health Organization, is for 2 years before you can say it's actually completely gone, and then you can stop doing these kinds of processes. The big problem, not surprisingly, is Afghanistan and Pakistan, particularly Pakistan, but this is really mostly on the border between Pakistan and Afghanistan. And why is this such an issue? Because it's not politically stable there. And in fact, vaccinators were shot. It was one of the very dangerous things to be doing in that part of the world. One of the things that has changed quite a bit in this vaccine elimination program has been to get as many local people involved in vaccination, and more and more and more of those, and that seems to have made a big difference. It used to be the people in white suits who come in and say, hey, you must take these shots. That didn't go very well. And so more and more local buy-in for these kinds of things. And so what are we going to do next? Oral polio vaccine has worked really well because of this spreading effect, but it has this downside, which is it can revert. And so the plan that the WHO has is to switch over from this oral polio vaccine to the inactivated polio vaccine. And in fact, that's in process right now, trying to get large stockpiles of the inactivated. But this, as you can tell, is a way easier way to vaccinate than this and needles and so on and so forth. And so the basic idea is when we get down to 0 cases, if there are any cases, then go in with inactivated polio vaccine and try and stop any further spread of disease. Okay, that's all I've got on the pathogenesis stuff, sorry. We're all clear on it? Clear enough that we can answer a clicker question on it? Yes, good. So, in the next few decades, the most likely source of poliomyelitis is type 2 wild, type 2 vaccine-derived, Nigeria, India, or Israel. Say, we're aiming for 100%. Again, feel free to talk about this. And then be quiet. I know it's Monday morning. You didn't get your coffee over at the biker breakfast.
Those are type 1 and 3.
Oh. I said B.
Kind of stupid. I actually feel kind of smart.
10, 5. What I should do is I can see the scores up here. I should just stop it when I think that we're in good shape as far as the 100% is concerned. But So vaccine-derived. So the vaccine-derived strains are the ones that are most likely. And one of the things that's happened in the last few years, those of you who listen to TWIV know about this, they've actually asked all the researchers to get rid of their type 2 so that there's no type 2 left around anywhere, so that can be gotten rid of. Any idea why I put Israel up here? Israel's close to Palestine, which is one reason, but it turns out there was a big outbreak, actually, of vaccine-derived polio a few years ago in Israel. They had about, I think, 20 or 30 cases, all of vaccine-derived. And so what did Israel do at that point? They switched to IPV, exactly. They stopped any of the oral polio vaccine usage there at that time. So, yeah?
When you see big outbreaks in tiny areas like that, is it somebody with the vaccine had a reversion and then spread it to people without the vaccine? Is that sort of what's happening, or is it the whole batch?
So it seems to be exactly your first scenario. So there's one person who's gotten the vaccine, that then reverts, and that then spreads to unvaccinated people in that area. Okay, other questions on the vaccine stuff? Okay, now the interesting stuff, well, from my point of view anyway. Structure, we already talked about this, so hang on, let me hide this. Structure of the poliovirion, pseudo T equals 3. What does that mean? It's Monday morning, I guess. So, multiple proteins that are put together in a quasi-equivalent structure, a lot like a soccer ball. Maybe a little hard to see here, but here's a 5-fold axis of symmetry. Where's our next 5-fold axis of symmetry? It's down here. Here's a 3-fold axis of symmetry. It's a lot easier to see down here. Here's a 5-fold axis of symmetry. There's a 3-fold axis of symmetry. There's a 5-fold axis of symmetry. And each of these different colors is, in fact, a different protein. Again, ridiculously creatively named VP1, 2, and 3. And 60 each of those. There's also a VP4. That VP4 sits on the inside of the capsid. And we'll see that VP4 is really important for, virus entry a little bit later on. How do polioviruses interact with the cell? They interact with the poliovirus receptor. Again, ridiculously creatively named. But interestingly, it doesn't actually interact with these 5-fold axes of symmetry. It interacts here in what's called a canyon around this 5-fold axis of symmetry. So normally, if you think about receptor binding, it's going to be the part of the virion that sticks out the most. That's not true for polio. It's actually next to the part which is sticking out as much. So we can zoom in and look at these individual proteins in a little bit more detail. VP1, 2, and 3 turns out have almost identical structures to each other. It's a little hard to see here. Maybe we'll turn the lights down. And they've got a central structure, which is this beta barrel structure. And the differences between VP1, VP2, and VP3 are these extra bits around the outside of that. And not surprisingly, here's our VP1 that has this extra loop that sticks up. That's the one that it's at the 5-fold axis of symmetry here. And each of these 5-fold axis of symmetry have these projections, which is where you can see that extra piece. Where the receptor binds is actually just off to the side of these VP1s here, so in this canyon structure. And when we looked at some of the entry processes, what we saw was that there are small molecules that can bind right here in this canyon, which are really good anti-polio, and particularly, more importantly, not so much for polio, but more for anti-rhinovirus or any of these picoRNAviruses. How does it get inside the cell? As I mentioned before, it's kind of analogous to what happens with bacteriophage. There's interaction with the receptor, right here, the poliovirus receptor, not here, but relative to this canyon right next to the VP1. Once you have this receptor virion interaction, there's a conformational change where VP4, again, which I mentioned is sitting on the inside, then basically makes a hole in the plasma membrane, and the genome comes into the cell. This genome has VPG that's attached to it that we'll talk about in just a second here. So binding to receptors, conformational change, release of the genome inside the cell. What is the receptor? It's the poliovirus receptor. Of course, this was not evolved to be a binding site for polio. It has a particular structure called the immunoglobulin superfamily structure. Why would one think that a receptor would have an immunoglobulin-like structure? What do immunoglobulins do? They bind to things. And it turns out poliovirus receptor, no, we're not entirely sure what the poliovirus receptor normally binds to, but clearly binds to poliovirus. And a lot of the receptors for different picoRNAviruses have this immunoglobulin, like fold to them. And I'm not going to ask you all the details on these, but immunoglobulins turns out to be evolutionarily a really good way to bind to things. And so it's not surprising that a lot of viruses have taken advantage of these immunoglobulin-like structures on the outside of cells. Why on the outside of cells? Because they need to bind signals that are coming from the outside. They need to bind nutrients to bring them in, etc. But it's not the only ones. There are also these what are called integrin proteins. Integrin does exactly like what you would expect. Binds something on the outside of the cell and brings it to the inside of the cell. Again, not surprisingly, viruses have evolved to take advantage of these particular processes. So what comes in? What comes in is this virus genome. The virus genome encodes one and only one protein, also known as the polyprotein. This is true for all of those picoRNA viruses. Some of them have different proteins here, and particularly the rhinoviruses that we won't really talk about too much. All these different proteins. There are basically two different sets of proteins that you have in these genomes. We've kind of talked about these before. You've got structural proteins and non-structural proteins. The structural proteins are also known as the capsid proteins. That puts everything together. And not surprisingly, it's VPs 1, 2, 3, and 4. Then you have all the non-structural proteins, which are involved in genome replication, but also in dealing with this whole polyprotein, because it's literally made as one big, ginormous protein. Those need to get chopped into individual smaller proteins before they actually have activity. So all of these proteins, VPs 1 through 4, have to get chopped into separate pieces before they can fit into that nice pseudo T equals 3 icosahedron. This polymerase protein, the 3D protein that we'll talk a lot more about later on, has to be separated from everything else in order to have its normal replication properties. A couple other things about genomes. Whenever you think about an RNA genome, what do you need to think about? Not what's coding, what else do you need to think about? What's going on at the ends, 5' end and 3' end. 3' end for poliovirus, which turns out to be different from some of the other picoRNA viruses, has a nice poly A tail. Great, wonderful poly A tail. This can get translated, absolutely no problem. On the other hand, the 5' end is very different. It has a protein bound to it, and that's the VPG protein. You may remember from the injection process, the VPG protein. It's covalently bound to the 5' end of the genome. And we'll see why that is a little bit later on. But what problem does that immediately pose? It can't bind to not so much the ribosome, but the translational initiation machinery. So if we look at translational initiation machinery, this is cellular, red, italicized, etc. Cellular translation. Messenger RNAs have caps. The eukaryotic initiation factors for translation, particularly EIF4E and 4G, bind to this cap structure and help the ribosome assemble. And the main thing about ribosome assembly and getting translational initiation is getting your initiator tRNA to this AUG site. So this is the cellular process. You have the small subunit of the ribosome together with the initiator tRNA that's associated with the cap structure through EIF4E and EIF4G. Once it's all together, then the ribosome chunks its way along till it finds an AUG, and you get translation starting. If you've got a VPG, you've got a protein bound to this end, then you can't assemble all of the translation machinery. You can't get translation. So there has to be another mechanism for doing this. Well, that's the internal ribosome entry site. The internal ribosome entry site basically provides a binding site for the initiation machinery in the absence of EIF4G and EIF4E. So it's just the small subunit of the ribosome together with the initiation factor 2, which is bound to that initiator tRNA. So this brings the machinery to this RNA, which is now the viral genome RNA, usually right next to the AUGs. There's not a huge amount of translocation that happens through here in order to get this translation to take place. Turns out that there are lots of different irises, 3 major ones that have slightly different structures, but all of them serve to bring the ribosome here to the appropriate start site. This is just one of those. A couple of things that I wanted to mention here. It's all about the secondary structures which form in your RNA. This should sound very familiar relative to what happens with the bacteriophage that are RNAs. It's all about the secondary structures. And a couple of things here that are quite important as far as getting this translation to start. Lots of different kinds of things here, different kinds of sequences, again, none of which are terribly important, but the real binding site for the ribosome is right next to this first AUG sequence, and will bind here and allow translation to take place, but it can only happen if you have disruption of these secondary structures. So you have this binding by the ribosome, those general eukaryotic translational initiation factors that'll bind to these different sequences. And again, I'm not going to emphasize them because they're different in the different irises. Close to the AUG, where you get translation that starts. And interestingly enough, there are actually 2 AUGs here. There's an AUG here, which is quite a long way upstream of the AUG for the polyprotein. So you have translation of a very short open reading frame, but that ribosome stays on the RNA, which would also should sound really familiar relative to MS2 and all of those bacteriophage RNAs, and then will be translated. So what is translated? It's a big protein. Yeah?
So that first sequence in between the first start codon and the second one, that gets translated?
In some of these enteroviruses, yes, not in all of them. So it varies. But once you have the ribosome associated, it stays associated with the RNA.
Is that in the ones that it gets expressed, is that a protein that just automatically gets degraded? That first?
So some of these peptides seem to be important for function, and in some cases they don't. So it seems to vary quite a bit. Okay, so what gets translated? Yeah, sometimes these short peptides at the beginning. But the main thing is this polyprotein. Single RNA, it's actually from the Flint textbook, but basically the same thing in ours. One big protein. That then gets cut up by 2 different proteases into smaller proteins. The first one of those is what's called the 2A protease, and then the 3C protease. That process, P1, is all of your structural proteins. P2 and P3 are your non-structural proteins. These two proteins are mostly going to be involved in this chopping up process, and the three proteins are all involved in genome replication. Particularly important, we already talked about the 3D protein. That's the viral polymerase. VPG is one of these three proteins that is going to bind to the end of your genome. How it binds to the end of the genome, we'll see in just a second here. So single RNA, polyprotein, split up by two different proteases. And these are viral encoded proteases. So they're in this viral genome. What does that mean about those viral proteases? They have to be functional before they get chopped out. So this 2A protease here, where's that? The red arrow, not seeing it here, there we are, has to be functional and basically cut itself out of this polyprotein. So what that means, between each of these individual eventual proteins, they've got to be floppy bits of protein that will allow that process to take place. And that gets back to Ian's question about, is this more efficient or less efficient to have these multiple proteins that, or have what would be differential translation or transcription machinery, I should say replication machinery, giving you a separate messenger RNA. And it turns out it's multiple different ways that you can do this. Here, you have to have something which is flexible enough that it'll have enzymatic activity even as part of a polyprotein, but you don't have to have these extra regulatory sequences. So it's a, and it turns out, we'll talk about these other RNA viruses which have separate regulatory sequences there instead. So lots of different ways to do this, skin the cat, as it were, or make the virus.
Can I ask a really simple question? You keep saying that it starts on when they give one protein. Does it mean everything encoded from P1 to P3 comes out as one single protein?
Yeah. So it's the ribosome that makes one big polypeptide, and that then gets chopped up into pieces. Okay. So what's our processing process? So let's see if we can do another 100% here. Huh? Ye of little faith. Processing of picoRNAvirus polyproteins due to the activity of RNA-dependent RNA polymerases, RNA-dependent DNA polymerases, DNA-dependent RNA polymerases, proteases or RNases. You didn't even have a peptide polymerase.
Don't do that. No.
You need a DNA-dependent DNA polymerase.
See, whenever I see the same thing, just slightly different wording, I always assume it's going to be one of those.
5. No 100% this time, but yes, it's the proteases, not these different polymerases. And one of the people up front I overheard saying is, it's got to be one of those different ones, the DNA dependent, the RNA dependent, so on and so forth. Those are just really easy kinds of answers to put together. I hate writing answers to these things. I know I should just make it really, really obvious all the time. That would make it easier, wouldn't it? So yes, it's all the proteases. In fact, the 2A and the 3C protease. The fact that I'm mentioning those letters a lot and numbers means you might want to think about remembering them. Just a thought. So how does genome replication take? Oh, sorry.
So the 2A is the only one, not the 3C that cuts itself out, right? Is the 2A 3C?
So let me just back up real quick here. The 2A just cuts here. 3C is over here. So 3C also is cutting itself out. But if you think about the translation process, this actually happens co-translationally. So as the protein is being made on the ribosome, this 2A protease will be made, and it will then cut here. Then more of this protein will be made. Then the 3C protease will cut down here. OK, so let's move forward here. Let's talk about genome replication. Genome replication of pretty much all picoRNA viruses is extremely similar. So this gets us to why you have this funky protein bound to the 5' end of the genome. One of the big problems with replication is, what's your primer? All polymerases, with the exception of the DNA-dependent RNA polymerases, need primers. They need an OH that they can extend from. And we'll see that there are some issues with this with lots of other RNA viruses. But with poliovirus and all of the other picoRNA viruses, they've got proteins covalently bound to the 5' end. And the reason they have proteins covalently bound to the 5' end is that that's the primer. It's a protein-primed replication process. And it has an OH, which can then be extended. What's the 3' end of the poliovirus genome? It's a poly A tail. What can bind to poly A? U's. So here, the genome is brought into the cell, gets translated into the various different proteins, and VPG associates with a membrane. And it turns out that lots of these RNA viruses are going to replicate on internal cellular membranes. And one of the ways, in fact, you can see virus infection in a lot of these cells is by seeing massive numbers of intracellular membrane vesicles. So also, it's a quite hydrophobic protein, binds to these membranes, and 2 U's get added to the VPG protein. Turns out that gets added here in the middle. We're not going to get into that in too much detail. But these 2 U's now can bind to the poly A tail. Now that bound to the poly A tail, the 3D polymerase binds to this and can extend all the way down to the opposite end of the genome. So this is great if you can get these U's associated with your VPG. How do you get the U's associated with VPG? Well, the other structure that I haven't talked about yet at the 5' end of the genome is this cloverleaf-like structure. And we talked about the cloverleaf-like structures for the RNAs, for the plant viruses. This is a very different structure. But again, it's a secondary structure that binds to the viral proteins, but also binds to cellular proteins. And there's this particular cellular protein, the poly-C binding protein. What does it bind to? Lots of C residues in this structure. And so it's a protein RNA structure which allows this polymerase to bind to the VPG and give it a couple of U's attached to it. And once that happens, now VPG can go over here, and you can extend your way down to the opposite end of the genome. Once you've made the negative strand, now, of course, you need to package positive strands for your genome. That turns out you don't need any extra proteins here. This negative strand RNA serves as a great template to make many, many copies of the positive strand. And this set of U's over here that you made the first time around now just gets copied into A's. So this is not a poly A polymerase process. So if you think about messenger RNAs, it's you get cleavage, you get extension by a poly A polymerase. All of the poly A's are encoded in these genomes. So every single one of those A's comes from the replication process through the 3D protein. So the 3D protein is the RNA-dependent RNA polymerase. When they solved the structure of it, they had kind of a big surprise. And what was that big surprise? What does this structure look like? It looks like a DNA polymerase. Well, that's bizarre. Of course, all of you know, finger, thumb, etc. from that class last year. But this RNA-dependent RNA polymerase, and it turns out almost all of the RNA-dependent RNA polymerases actually are a lot like DNA polymerases. They just use RNA instead. Now, we could have guessed this already. Why? It's because all of these polymerases need primers. DNA polymerases all need primers. So it's not surprising that these things have very similar kinds of structures. And we'll see this again and again when we talk about these different kinds of structures. The big exception to this is that T7 DNA-dependent RNA polymerase that has a very different structure, even though it's a single subunit. Yeah?
Do they also resemble DNA polymerases in having better proofreading activity, or is it mainly the primer?
So the question is having good proofreading activity. So a lot of the proofreading activity comes from separate domains of that RNA polymerase, the DNA polymerases. These do not have those proofreading domains. So if they make a mistake, there's no way that they can chop that back out. So it's not there. So they don't actually have good proofreading. Okay, so that's all for replication of our genome. Last thing that I wanted to talk about was the assembly of these virions. Again, it's all made as a precursor protein. This gets cleaved into 3 separate pieces, VP1 and VP3. And what the heck, VP0? What the heck is that all about? We'll get back to that in just a second. But VP1 and VP3 get assembled into these trimeric structures with VP0. Well, that's VP0. VP0 is just VP2 and VP4 that are still connected to each other. You make each of these triangular faces. You're going to need 20 of them for your icosahedrally symmetric particle. Those come together, make these procapsids, which again should sound really familiar, really similar to what's happening in the bacteriophage capsid formation. And then when you have this final structure, then and only then do you have proteolytic activity, which now gives you an active virus particle. If you remember, it's the VP4, which is now sitting on the inside, that has to undergo the conformational change in order to make the pore through the membrane. If it's still attached to VP2, it can't do that. So this is another example of how you can have a metastable state. It's really stable until you have that last proteolysis and that last cleavage that actually takes place. There's one more thing as far as proteases is concerned that's very important, and this gets back to the irises. So internal ribosome entry site, you don't need all of the cap binding and bridging proteins that you normally need in cellular translation in order to get translation to take place. So one of the activities of the viral protease is actually to cleave that cellular translational initiation protein. And what that means is that all the cellular cap-dependent translation gets shut down completely when you have a poliovirus infection. And that's basically shown here. You have infection of a cell with poliovirus, cellular translation goes way down, but then you have IRES-dependent translation from your new genomes that have been made by the poliovirus. So this is where you get all your translation. So if you just look at radio-labeled cells, they end up just making all of these different viral proteins. There's also inhibition of the cellular DNA-dependent RNA polymerase and a block of vesicle transport, which means you accumulate lots of vesicles inside the cell. So guest lecture on Wednesday, Dr. Hirsch from OHSU definitely be able to post his slides. Whether he wants to record or not, we'll see.